Hello, welcome back to the magazine. Today we are going to discuss about chapter 4 biological molecules under IGCSE Biology. Before start the lesson, I have a small news to you. So this is your first time you are watching, so please subscribe me and click the bell icon. Then once I upload the videos, you will notified, as well as share the videos with your friends or with your students, because in the magazine content I have shared more videos. I will share more videos regarding biology, chemistry, as well as there is a podcast series which is related to the IGCSE exam. How do you need to select the exam codes, what are the criteria, like that we are discussing about that also. As well as another important news is I am discussing the past papers as well. So I'm basically focused to the most recently past papers and what are the predictions for a next year examination or a next exam series. So these are the content I'm going to share with you through the magazine. So please follow me and share it with your friends, as well as if you have any questions regarding IGCSE or biology or whatever, please put the comment. I will definitely answer to you. So let's start it. Here you can see a short note, a mind map which is related to the test. Those are including in your textbook. You have to study 4 types of test. You need to know about the sugar test, starch test, protein test, and oil or a fat test. So first of all, let's see how can we identify sugar. So especially carbohydrate test, there are 2 types of test. First one is sugar test, another one is starch sugar or residue sugar they are calling. So for a sugar or a residue sugar, we have to check. For that we need to use some indicator we are calling Benedict solution. So this Benedict solution is blue color. When we add to the residue sugar solution, and we have to heat that. Once we add, at the moment the solution residue sugar solution and the indicator mixture will get orange or orange yellow color. While heating, it would be changed green. After that, once all reaction has completed, it converts into the orange red or a brick red color. So if you receive brick red color, that means that solution, that sample is including in residue sugar. So let's move to the starch test. How can we identify starch? So for a starch identification, we are using iodine solution, which is like yellow orange color. So once your solution including starch, that indicator convert into the dark blue color. Next one is protein test. So in here we are using biuret reagent, which is the color is like blue. So once you add it to the protein solution, or if your solution protein available in your solution. So blue color convert into the purple color. So this is the indicator. You can identify protein present or not. Last one is fat identification. So we are calling for this test emulsion test as well. So here there is some procedure. What you want to do, oil mixture or the mixture that you want to test, we have to add for that ethanol and mix thoroughly. Then the oil, you can see the whole oil, it will be dissolved in the ethanol. Once all dissolved, you have to add some water into the same solution. Then again you have to mix. Once you mix, you will see some oil drops. Those are transferred to the water layer from ethanol layer. That water layer will be converted into the milky color. So transparent water converted into the milky color with oil globules. So this is the indicator. You can identify fat or oil that are present or that are available in your solution. These tests you have to keep in the mind, because every past papers I saw these question, this kind of testing questions are included. So I highly recommend to follow my paper discussion videos. So you will see there are so many questions which are related to the test. So study this and follow this channel, as well as if you need these mind maps or short notes, let me know. Put in the message in the comment section. I will share all resources without any charges. All are free. So let's move to our another second part to study about the structure of DNA. So in this slide you can see the mind map which is related to the structure of DNA. So the smallest part or smallest molecule is nucleotide, which is build of the DNA or a structure of the DNA. This nucleotide formed by using a carbohydrate, a phospholipid or a phosphate and kind of a protein. So these are connected to each others, formed a nucleotide, and this nucleotide, the different kind of nucleotide gathered together, DNA, one side, one strength will create, or one strength will be formed. So in the nucleotide there is a protein, and for this protein which is connected to the bases. So A, G, T, C are the main 4 bases. A means adenine, G means guanine, T thymine, C cytosine. So a special thing is this kind of bases pairing each others. A, adenine based with only adenine combined with only thymine, cytosine combined only with guanine. So when discuss about the structure, we already know that is a double bond, including double bond, and it is a helix like a ladder. The ladder which is rotated like helix. So these are the main points we have to know about the DNA structure. This DNA structure we are using to transfer the genetic materials to generation, as well as the DNA structure will be help to create a protein. or a protein synthesis, the main structure is DNA. So in the DNA structure, it is there are two strands which are connected by the hydrogen bond, like double bond, as well as these two strands which are arranged as a helix, which are called double helix. And those double helix which are connected to the edge by using base pairs. There are four types of base pairs: adenine, guanine, thymine, cytosine. Adenine combined with to the thymine, cytosine combined with to the guanine. So the structure, the smallest molecule of the DNA, we are calling nucleotide. So today I have covered your chapter 4 IGCSE Biology. So stay with me if you have any doubts related to this lesson, this chapter, or any other related to the biology, or if you need my short notes or a study material, so put the comment in the comment section. I'm ready to share my whole documents with you, as well as if you have any questions, I'm ready to answer for those. So again, thank you so much to watching my channel and watching the video and joining with my channel. See you again as soon as possible. I will upload chapter 5. You will learn more about those things as well. Stay with me. Sharing the past paper question discussion as well. See you soon. Bye bye.